For our next video clip, we're going to talk in a little bit more detail about the structure of viruses and take a look at some of the diversity in virus morphology. So we've heard the term morphology before. That refers to the shape and the structure of something. And so this is going to be very different when we're thinking about viruses because they are acellular and much different from any other type of microorganism that we talk about in this class. And then even among viruses, there are many different types, and no doubt many different types of viruses have never even been discovered yet by microbiologists. Okay, so this is kind of the generic basic virus over here. The simplest type of virus will have its genome packaged up inside a protein shell called a capsid. Okay, that's the simplest type of virus that you can have. Now, the capsids can have different shapes. So this is called a polyhedron or sometimes an icosahedron, and that is built, all those little spheres in there are capsid proteins. So there might be one kind of capsid protein, then they all get together and assemble into this geometric shape, or there may be more than one type of capsid protein that assemble that type of structure. But this is a pretty common shape among viruses. Now the capsid does not have to have this shape. Another common shape is this, a helical capsid like you see over here in this virus, which means the genome will be in the center and then it's coated with these proteins and you have an overall spiral shape. And then there are some other variations from there, but that shape and this helix type shape, those are the most common among viruses. The capsid, the purpose of that is to protect the genome, so the genome does not wind up getting degraded while the virus is traveling from the host cell that it left to find a new host cell to hijack and infect. Then some viruses have additional layers, and a major layer would be the envelope. So some viruses have envelopes and some do not. So this is an example of a virus here. This outer layer is an envelope. Here's another one over here with an envelope layer. And the envelope is really phospholipid bilayer membrane that has been stolen from the host cell. And we'll see how that happens. It occurs when virions are released. So this term virion, when you see that used, that is referring to a fully assembled viral particle that is capable of infecting a new host cell. So it's not partially built, it has been completely built and assembled together. So sometimes those are called virions. So the envelope, not all viruses are enveloped, and we will talk more about enveloped versus non-enveloped viruses as we move through the rest of the course. It especially comes up when we start talking about disinfectants and antiseptics, because as it turns out, enveloped viruses tend to be a little bit wimpier. You would think maybe they would be hardier because they have this outer shell, but that envelope is a phospholipid bilayer, and it's fairly easy to dissolve that layer and destroy it for many enveloped viruses. So this will come up again. And some viruses will have intermediates in between, and they're showing that to you here where you kind of see this light green layer on that virus and the other one over here. That's an additional, like a middle layer protein coat, sometimes called the matrix, and that exists on some viruses and not on others. Another feature of enveloped viruses is that they will have proteins embedded in the envelope. So just like you have membrane proteins sticking in phospholipid bilayer cell membranes, you also have proteins embedded in the envelopes of viruses. And these are actually glycoproteins, which means they're proteins with sugars attached to them. All right, so what are these glycoproteins used for? Many of them are involved in attachment. So a virus has to attach to a host cell in order to enter that host cell. And host cells have receptors on their surfaces to which those viruses will bind. And so that binding usually occurs through one or more of these glycoproteins that are sitting in that envelope, if you're thinking about an enveloped virus. And this envelope virus that you see here, has more than one kind of glycoprotein in its envelope. This one looks like it has just one type. So there's a lot of variation out there among different kinds of viruses. And you can have an enveloped virus with a helical capsid on the inside. You can have an enveloped virus with a polyhedral capsid on the inside. You can have non-enveloped viruses with polyhedral capsids. You can have non-enveloped viruses with helical capsids. There's just a lot of variation. This is a figure from your textbook, which is trying to drive home give you some perspective on the relative sizes of viruses. And we did this before back earlier in the class with an interactive activity. But this is showing you. And here's in green or greenish blue here. You've got some example yeast cells. So they're trying to give you yeast or single-celled fungi. And here's an E. coli bacterial cell about 2 micrometers long. Here's an individual streptococcus bacterial cell about 1 micrometer in diameter. Rickettsias are even smaller types of bacterial cells, and many of those, even though they're bacteria, many of those are intracellular parasites as well. So they've actually got to go inside a host cell to multiply, but they're still a cell and a bacterial organism. They're not a acellular virus. Rickettsia Rocky Mountain spotted fever, for example. If you've heard of that, that's caused by a rickettsia type of bacteria. And then here are your viruses. So quite a wide range of sizes. The largest known viruses are called viruses, and they actually infect algae. So algae are their hosts, and some of those are 450 nanometers in diameter, which is almost half of a micrometer. And a micro is something that's 1 micrometer in diameter, like a streptococcus bacterial cell here. You can see that with a light microscope in a typical laboratory. So these are getting close to the limit of something that you could see with a light microscope like we have in our labs here at Calhoun. But this would look like just barely a speck if you were doing that. And here's herpes simplex viruses, or herpes viruses, about 150 nanometers in diameter. Rabies virus, 125. Here's HIV, about 110 nanometers in diameter. Influenza, and so forth and so on. Some of your smallest viruses include polio virus and yellow fever virus, which they're getting so small, they're not much bigger than a hemoglobin molecule like you would find in your red blood cells, the protein molecules that bind oxygen gas and help transport it throughout the body and the blood. So that was just some size perspective. How do we classify viruses? How are they grouped together with their other relatives? And so a lot of this is based on, traditionally it has been based on the viral morphology. So you take a look at the overall virus particle, and what kind of covering does it have? Does it have an envelope, or does it just have a capsid? Then you look inside the central core of the virus, and what do you have there? You've got the nucleic acid genome, and so is it DNA or is it RNA? And notice here you've got molecule with a possibility for pluralization. So some viruses have more than one DNA or RNA molecule inside that central core. And then some viruses have matrix proteins, which are like a middle protein layer between the capsid and the envelope. Some viruses do package enzymes that they need in order to begin the process of replication when they get into a host cell. So all those things are the traditional ways that viruses have been classified. It's based on these characteristics. You look and try to put them together in groups of related viruses. Now today, because we're getting so sophisticated with our genetic analysis techniques, and viruses tend to have pretty small genomes, so anywhere from a few thousand base pairs, up into the hundreds of thousands of base pair range. But that's pretty small compared to a living organism, and so we have the technology now to sequence the genomes of viruses pretty quickly. And when you compare the genetic sequence of one virus with another virus, the more closely related they are, the more closely their genetic sequences will match up with each other. So a lot of viral classification is based now on genetic similarity and not so much virus morphology. So here's a table in your textbook, which again is just trying to emphasize for you some of the diversity out there among viruses. And this is more specifically zoning in on viral genome diversity. So viruses can be either DNA viruses or RNA viruses, and that's referring to the genome that's packaged up inside the virus. Is it a DNA molecule or an RNA molecule? All right, and then among your DNA viruses, most of them are double-stranded DNA viruses. That means the genome would be just like the genome of a living organism. It's going to be a double-stranded DNA helix. But there are some that have single-stranded DNA. An example of those would be the parvoviruses, and maybe you've heard about dogs getting parvoviruses. There are a number of different types of parvoviruses that are all related to each other, but they can cause very different kinds of infections in their host. For example, parvovirus, canine parvovirus causes a deadly illness, and your dog gets vaccinated to help prevent that. In humans, an example of a parvovirus infection would be erythema infectiosum, which is a skin condition. Little kids get it sometimes, and it looks like their cheeks have been slapped because it's the viruses are infecting the skin and triggering inflammation there at that location. So, those are closely related viruses, but one type causes a deadly illness in dogs, and another type causes a much less serious infection in humans. Many viruses have double-stranded DNA genomes, herpes viruses, for example, but many others as well. Then among your RNA viruses get classified according to some characteristics of the RNA genome that gets packaged up inside the virus. So some of them are single-stranded. There are some examples of viruses that have double-stranded RNA genomes. And typically we think about RNA as being a single-stranded molecule, but like I said, the viruses violate these rules sometimes, and there are viruses with double-stranded RNA genomes. And then there are some viruses that have single-stranded RNA genomes, but they get converted into DNA in their host cell, and we'll spend a little bit more time talking about that later. Then on this table, when you're taking a look at it, you'll see some of your single-stranded RNA viruses are classified as having positive polarity, or they're called plus-strand RNA viruses. Some are classified as having negative polarity, or they're negative strand RNA viruses. And what that means, and we'll see a little bit more of this later, if an RNA virus is considered to have a plus-strand RNA genome, what that means is when that RNA genome enters a host cell, the host cell sees it as a messenger RNA molecule. So messenger RNA molecules are the ones that attach to ribosomes, and then the ribosomes read them and translate a protein. Negative strand RNA genomes, on the other hand, when they come out of a virus and they go inside the host cell, they're actually complementary to messenger RNAs. So they actually have to be converted over into the plus strands that can be treated as messenger RNA by the host cell. So it's all about whether or not that genome can function as a messenger RNA inside the host. Okay, so that was just kind of giving you a little bit of a overview on virus morphology and the diversity that's out there. We'll be moving into the third video clip, where we'll start talking about some general aspects of viral replication. All viruses have certain steps they have to fulfill in order to be reproduced, but then they take off and have very different ways that they accomplish those strategies, and so we'll be taking a look at that in more detail as we go through more detailed life cycles of some example viruses when we get to the fourth video lecture.